So, hello, nice to meet you. My name is Tom Parks. I'm a clinical research fellow from the University of Oxford and the London School of Hygiene and Tropical Medicine. And perhaps it's worth explaining that my background is a clinician in infectious diseases, and I come from a human genetics research background. So this is my first time attending this meeting, which is really fantastic and exciting. But I may have a slightly different perspective on some of these issues. So bear with me, and then you can throw some questions at me and challenge perhaps some of what I'm going to say. I'm going to talk to you a little bit about group A streptococcal disease, which is the disease I'm interested in and work on, and the group of diseases I'm interested in and work on, and then talk mostly about our work on host genetic susceptibility to rheumatic heart disease. And then at the end, we'll talk a little bit about some of the ways we can move forward in this with using novel long-read sequencing approaches and alternative way of looking at some of these problems by integrating signals from different diseases to get at the problems. So, group A streptococcal disease is not something that's particularly thought about in the antibody field, but I'm going to try and convince you that should change. So hopefully I've got a pointer here. So the group A streptococcus, or Streptococcus pyogenes, is the common bacteria that causes strep sore throat. And strep sore throat, superficial infection, is clearly a sort of common mostly childhood infection, and it's something that will occur to most individuals on their way to adulthood. Actually, in the tropics, the major manifestation is pyoderma and impetigo, so streptococcal infection on the skin. And in some parts of the world, those are actually sadly normal parts of growing up in childhood, where impetigo infection is common and will occur in sort of 10, 20 or more % of the children at any one time. And there are these two rather distinct complications of those superficial infections that occur in some individuals. So firstly, our invasive and the toxin-mediated diseases. The invasive infection is where the infection moves from the superficial site into deep tissues, often in this disease in soft tissue infection, but actually it can occur in a variety of locations. And particularly toxic shock, which is a complication of that, where the immune system is hyperactivated. These are associated with very high mortality, and as a clinician, the clinicians in the room will know these are some of the sickest patients you ever see. But there's some other important manifestations here, particularly puerperal sepsis, which is childbed fever. This is the classic cause of the sort of childbed fever of the 19th century. But actually that remains a problem in the UK and elsewhere because of the challenges of delivering good high quality obstetric care. Then on the other side, we have these post-infective manifestations. So the infection has cleared, and then 2 or 3 weeks later, and in fact it's probably more indolent and chronic than that in reality, you get this abnormal autoimmune, for want of a better term, response, manifested as acute rheumatic fever, which is an acute febrile disease, rheumatic heart disease, which is a chronic inflammatory disease of the heart valves, where you get scarring very specifically of the heart valves, which causes the heart to fail, and you get complications and death as a consequence of that, or a kidney disease called acute glomerulonephritis. And together, these are the 6th leading cause of death, infectious death globally. So it's a similar number of deaths to the likes of malaria, which I'm sure is much more on your radar, but also the major cancers, so breast cancer, prostate cancer, a similar number of deaths to this. The problem is we have very limited control strategies at the moment, and we don't have a vaccine, and so actually, particularly rheumatic heart disease, which is by far the biggest cause of death amongst all of these, remains unchecked across much of the developing world. So, I've been interested for a few years now in trying to understand host genetic susceptibility to these diseases, and that's about trying to understand why one specific pathogen causes such a range of different disease phenotypes. And we do this 2 ways, one by looking at rheumatic heart disease using genome-wide approaches, and then invasive group A streptococcal disease using a combination of genome-wide approaches and exome sequencing. So my doctoral work was in the Pacific, and we set up a large sample collection across the Pacific region with case control studies in New Caledonia, Fiji, and Samoa, with a total of over 3,000 individuals from these different sites. And our cases were individuals with rheumatic heart disease diagnosed by ultrasound, so it's a very specific diagnosis, and our controls were members of the general population. And as you might imagine, the Pacific has very diverse ancestry, and I'll come on to how we dealt with that in a moment. And we used a combination of directly genotyped genotypes using an array, a genotyping array called the Human Core platform, which gives you about 500,000 genotypes across the genome. And then from that, you impute up to 5 million variants. And we used a combination of 1,000 Genomes platform and our own Melanesin platform. So the population structure and the diverse ancestry is key to all of this. So there are 3 major populations within this: the Melanesins, Polynesians, and Fijian individuals. And these are principal component plots summarizing that variation. So our analysis was stratified into those population subgroups, and all of the results I'm presented are meta-analyses of these different population groups. So here's the kind of headline summary of this, the published work, and then I'll come on to show you some unpublished follow-up work in a second. So we do a discovery analysis in the Melanesins, and then we replicate the findings in the 3 other populations. And here's the major signal, which is in the immunoglobulin heavy chain locus at genome-wide significance. Before we go on to talk about that in more detail, here's the HLA locus, which was the major candidate for this disease before this work. And so if we zoom in on the IGH locus, there's 108 variants at genome-wide significance. So that's a sort of stringent threshold used for statistical significance in genome-wide association studies. And if we zoom in on that using a Bayesian fine mapping approach that asks which of the variants explain 95% of the signal, you have these 174 variants, of which 6 are coding, and 5 of those coding variants fall within the V461 heavy chain. So those 5 variants, here's a model of V461. This is the heavy and this is the light chain, and the spheres represent the amino acid changes. This proline to alanine substitution here is of particular interest because it sits on the interface between the heavy and light chain. To try and confirm or refute this, we undertook some Sanger sequencing of the V461 locus. So we amplified a 1.5 kb segment and then sequenced within that 473 bases. And here's one of the some chromatograms showing the IGH V46102 allele substitution, that proline to alanine change. And by doing this, we did this sequencing in 339 individuals and imputed that information back into the wider data set. And we could increase our imputation accuracy up to 0.94 as a standard measure of imputation accuracy. And so with that information imputed back into the data set, this is the signal across these different populations, and we have a combined odds ratio of 1.44 per copy of the minor allele, the 02 allele, and this very strong consistency across these different populations. So there's one other detail that I don't get to talk about much because it's of less interest to most people, but I hope it will be more interest to this audience. So within that V461 locus, as you might expect, it was more complicated than we would have thought initially. And so actually there was a novel allele in there. So in addition to the 01 and 02 alleles I've mentioned, there was this 09 allele with a 6 base in-frame deletion. And actually what that does is that converts the sequence to exactly the sequence of V459-01. And of course, our initial anxiety was, are we just amplifying V459 because of non-specific binding of our primers? But almost certainly not, because these are the sequences, and the gray illustrate where the sequence that we amplify matches to V461, and there's only one position where it matches to V459. So what we've got here is a novel allele in a disease-associated locus, which actually is probably duplicated from the neighboring gene. These are things that are of interest to us because we're interested in the locus, but actually here it is existing in a disease-associated locus. So just to summarize that section and the published work, this was quite a big deal for rheumatic heart disease because this is the first genome-wide association study of rheumatic heart disease and really a kind of step forward in terms of understanding the immunogenetics. But I think hopefully it's also a step forward for the immunoglobulin heavy chain locus field and the immunogenetics field more broadly. But there are caveats. So we only actually have 16 genotype variants within IGH. We have 4 within the region that, well, 4 at suggestive significance within the region that we're associating with disease. But essentially we get lucky here because there's wide LD in the Pacific Islanders and we happen to pick up this signal. If the genotypes had directly genotypes had been distributed elsewhere within IGH, we'd have almost certainly missed this. So I think there's a lot more to unpick here. So I'm now going to talk a little bit about some unpublished work trying to replicate and refine the signal. And we've got 3 data sets we've used to do this. So the first is a case control study we've undertaken with collaborators in northern India, where we have a further 540 cases and 400 controls. Then we have individuals from the UK Biobank study. If you don't know about the UK Biobank study, it's worth knowing about. This is a very rich cohort of 500,000 individuals from the general population for which there are extensive genotype and phenotype information. And within that, because it's such a large population, there's a small number of individuals with mitral stenosis, and we include them, dividing them into Caucasian and non-Caucasian individuals. And of course, there's plenty of controls, so we can pick very well-matched controls based on a number of variables and use a 1 to 10 case control ratio. And then finally, and I've only just had these data for literally a couple of weeks, but there's the RHD Jain consortium, which is another genome-wide association study of rheumatic heart disease in sub-Saharan Africa across 8 cases and controls, 8 countries, recruiting cases and controls from all of them. And there's a large number of Black African individuals and then mixed African, mostly from South Africa and the South African colored population. So, if we then combine those different populations using a trans-ancestral meta-analysis, this is that locus again, here's the signal. If we just look at the first 7 populations, so leaving out the RHD Jain data, the 2 African populations, the signal pretty much stays where it is over V461, but with an eye of faith, I'm going to try and convince you that it's starting to move down the locus a little bit. If we add the African population, again, in a fixed effect meta-analysis, the signal remains there pretty clearly in over V461. And I mean, it's reassuring that actually, despite the fact this African study is an entirely independent genome-wide association study of a large scale, they replicate clearly this finding. But that's not particularly helpful because all it is doing is sort of pointing to the same things we found. So if we switch to a random effects analysis, which looks for consistency across these different populations in a more conservative manner, the signal really does change, and it changes down to V169. And I think that's helpful because, as I'll try and convince you in a second, we were never totally satisfied with this explanation of the findings. So there's the signal over V169, and here's some forest plots of those 2 signals. So in a fixed effects analysis across the 9 populations, here's the original SNP we reported in our paper. The signal's clearly there in the 2 African populations. That's reassuring, but it's rather unconvincing, this sort of heterogeneity. But if we look at the random effects model, we find this much more consistent signal. The p-value for heterogeneity here is about 0.9. And this SNP is very interesting because rather than it just being a sort of random SNP without annotations, here is a SNP that massively determines V270 expression with a p-value of 10 to the minus 30. And it's got all sorts of annotations in B cell types. It's a transcription binding factor site and a promoter flanking region. And so I just wonder whether we're starting to get a little bit closer to function with this. But I think there's lots of uncertainty, and so we don't really know what the effect is. We originally proposed that the effect was coding, and I think that's because in Post-genetics, there's a big bias towards coding findings, and the signal did appear to be clearly coding. Perhaps actually there's something more to do with regulation or expression, or something to do with copy number variants, or more than one of these, and I think that's quite possible. I think in the Pacific Islanders, V46102 clearly tags this susceptibility signal. It's just we don't yet know which of these is the underlying causal variant. And I think there's a lot more to understand about the effects on repertoire and then the downstream function. So just a couple more brief things to sort of try and tie this up and show you where we're going with the work. So I've been working a lot with Corey Watson, who sadly isn't at the meeting, but I think he's probably listening online. And many of you will be familiar with this fantastic work he's doing with PacBio long-read sequencing. And this approach, I don't claim to be an expert on it, but is about isolating long segments, 5 kb segments from the IGH locus, enriching using a capture assay for IGH enriched fragments, and then sequencing them using PacBio. And here's the way Corey's lab calls the data. So it's a haplotype approach, so that the reads are used to phase the data, and then the reads are assigned to each haplotype. And that means you get these very rich, long, extended haplotypes across the locus. So this is the data from one of our samples, and actually deliberately this is a sample with that novel allele, the allele we referred to as IGH46109 in our paper. So here are the non-partition reads, and then you can partition the reads by haplotypes. And as an example, one of the first things you can see is this individual is homozygous for several of the known copy number variants. This is sort of mind-blowing in the sense, remember I said we had 16 variants across the whole of the locus. With imputation, we got up to a few hundred, but we were never quite sure about the accuracy. Now we have thousands of variants across the locus, including accurate calling of the copy number variants and the alleles from all of these gene segments. Here's the region of interest around V461. And if we zoom in on that, we can clearly see that there are 2 different haplotypes over V461 and only one over V459. And there's, if I should have put a circle around it, but there's that deletion clearly situated over the 461. So that's that novel allele that we called 09. Of course, this is what it's closest to in IMGT, which is an V459 allele that's almost certainly been misattributed to V459. So just, if I've got time, just to finish on trying to integrate these different group A streptococcal traits. And I suppose the long-read sequencing is a sort of one approach using a kind of data-orientated way, and another way is to ask a different clinical question. And one of the sort of burning clinical questions we'd had, or I'd had right at the beginning of this is, are there genetic variants that predispose to one of these group A streptococcal traits but protect you from the other, and vice versa? So we've got a small genome-wide association study in invasive group A streptococcal disease. Note that number of cases. So I'm not pretending by any means that this is definitive, but it's a sort of first look at susceptibility to invasive group A streptococcal disease. And these are cases of invasive group A streptococcal disease from the UK, group A streptococcal empyema, which is a group A streptococcal infection in the chest wall, usually after influenza infection, childhood bacteremia in Kenya, and then a small number of invasive infections in the UK. And what we're looking for here is a general signal of susceptibility across these different populations and traits. So of course there'd be heterogeneity between these different disease traits, but the hypothesis is that there'll be variants that drive susceptibility to all of them. And the data I'm going to show you uses a compare and contrast meta-analysis approach, which is basically using the Z-scores to say, can we detect alleles that appear to be specific to one trait or another, or have a shared effect or an opposing effect. And we've done this just for the IGH locus, or we've done it genome-wide, but I'm going to show you the data for IGH, which I think is the most interesting. So using this approach, we can say there's a bunch of variants here which are RHD specific. Those are the ones I've sort of already talked about. But is there anything going on for the other shared or specific variants? There's almost nothing that's invasive specific, but that may well be power rather than anything else. There are some effects that appear to be related to shared effects, and they sit much closer to V169, and again appear to have regulatory effects, as I'll show you in a second. And then we've got a problem with power here, but you just start to wonder whether there are some opposing effects as well. So to sort of give you a bit more idea about what they're doing, I said these have regulatory effects, I believe. So here's the RHD specific signal. It strongly associated with less V270. By the way, these associations with expression I'm talking about are from whole blood RNA-seq studies rather than repertoire studies. So a key thing is to do this in repertoire specific work rather than just with whole blood RNA-seq. But V270 and V169 are clearly reduced if you have the minor allele for this RHD specific variant. Then there are these variants that go in the other direction, which increase your V270 and appear to protect you from both of these two diseases. And then there's another coding variant, but I think it's actually got a regulatory rather than coding function, or at least it's tagging a regulatory effect, which is the 03 allele from V366, which strongly affects multiple gene segment expression, and it increases your susceptibility to rheumatic heart disease and protects you from an invasive group A streptococcal disease. So to try and tie this together, I'd argue that group A streptococcal disease by itself has a major burden of disease and is important, but I think hopefully for this audience in this field, it's a key, it shows the sort of key relevance of the sort of work related to germline and repertoire to disease and public health on a global scale. We've been able to follow up our initial work by replication in independent African and other studies, and the fact we replicate in that large African studies is really reassuring to the findings. I think resolving the signal and understanding function is difficult, and it's going to take a while. It's not surprising that 2 or 3 years after doing the initial work, I can't stand up here and tell you this is the mechanism. But I think we have to get better at understanding the germline locus, which maybe that targeted long read sequencing is a way to do that, and understanding the relationship between all that variation we can now capture and the expressed repertoire and the function in terms of serological responses to group A streptococcus is key, and I think is the way forward, and certainly what we're now starting to work on. So there's lots and lots of people to acknowledge, as you might imagine, including the funders for all of this and the various groups who've worked on collaborating these data sets. I'd particularly like to thank Kate Auckland, who works with me on all this in Oxford, and Corey, who's, of course, much of his, the targeted sequencing approach was his work, or in collaboration with him. Thank you very much.
Tom? Questions?
So I mean, I don't know anything about these diseases, and you explained that the rheumatoid heart disease and the strep are somewhat related physiologically or something, or?
So they're caused by the same infection,
bug.
the same bug,
oh, okay.
the same bug.
Okay.
Yeah.
But
yeah, so the same, so I mean, and there's variation within the bug, as you might imagine, but not in a way that explains it. This same infection that you get in the throat, some individuals, most individuals will get better, a minority will get this invasive infection with high mortality, and then others will get this chronic inflammatory disease of the heart valves.
Yeah, I mean, this is so exciting, and I'm just thinking, well, is there the next disease then that you would obviously look at, or it's just
I mean, my bosses back home tell me to contain my work rather than expand it. But joking apart, I mean, I think this is, yes. I mean, I think this, as you and Corey have described, this is totally neglected across the bulk of genome-wide association studies. That's largely, as most of the audience will be aware, because of the extent to which these variants are not tagged on genotyping arrays. But I think we can start to change that. So I think across a range of infectious diseases, this is relevant. I think it's certainly relevant in diseases, infectious diseases where you get variable antibody responses, of which there are many. So malaria and TB being obvious examples where the antibody responses are variable and difficult to study, so people only study them to an extent, and the germline is likely to be key. I think across autoimmune diseases, it will be really important, and the UK Biobank dataset will help us do that. So the UK Biobank genotyping array has a much richer set of variants in IGH. It's far from perfect, but if we can integrate some of the long read sequencing data with the array data and do imputation, I think there's an opportunity to at least generate some initial data and signals. But I think ultimately we have to go back to some of the original GWAS data sets and persuade the investigators to get us DNA. We need an array that better captures this, and I think we'll start to get signals. And I think get at mechanism and the therapeutic antibodies.
Very nice study. So in terms of reactivity, do you have any idea whether the signals that you are seeing, whether this is correlating with epitopes in terms of the polysaccharide, or do you think that this binds to protein antigens? And an extension to this, is there is the epitope for the lens field A actually known?
So there is a proposed epitope in the literature, which is the M protein, but I think much of the work that underlies that doesn't really stand up to sort of scrutiny of kind of modern approaches, particularly given some of the non-specificity of the T cell response. So you do find antibodies against M protein in the sera of rheumatic patients with rheumatic fever, and you find antibodies against myosin, the sort of major cardiac protein. But I suspect that's got more to do with epitope expansion in a sort of autoimmune process rather than being the underlying, the causal antigen or epitope. I think in terms of what the epitope's driving these patterns, we don't know. I mean, we can speculate a little bit, and there is some work showing that these heavy chains themselves, V3 and V4 heavy chains, are themselves targets of autoantibodies in sera of rheumatic fever patients. So I haven't shown you that, but we have some new findings in complement and the complement factor H locus. And so I think actually there could be something to do with immune complex formation and other binding between antibodies that perhaps could explain some of the function.
Yes, so I wanted to kind of related to what Christian was asking. So what about serotypes, different serotypes of group A strep? And I'm just thinking the only example I know of really of genes being missing and therefore not having a good response is against carbohydrate, Haemophilus influenza. So I'm just wondering, do you have any feelings about the anti-carbohydrate antibodies and looking at those, especially, are there particular serotypes of group A strep that are involved in invasiveness, for instance, and so forth?
Okay, so the carbohydrate antibodies are important in a response to group A strep. I mean, there's lots of carbohydrate on the surface of group A strep. It's very, very similar to the epitopes and moieties are very similar to human tissue, so that there's bound to be some cross-reactivity there, but those are difficult to study at the moment, and we've got some ideas about how to do that, but purifying the carbohydrate is extremely difficult, and I think until we've got expertise in that, do you want to follow up? You're looking at though that's not satisfactorily answering. Okay, so the carbohydrate will be involved, but I think we need to get further with that. In terms of serotypes driving the disease, if you read the sort of textbooks, there are specific rheumatogenic serotypes and also nephritogentic serotypes associated in the disease, that's almost certainly nonsense, actually, and reflect the sort of epidemiology of the disease in a temperate environment where you get epidemics. So you get epidemics of a specific M protein or M type in the population, and then a few weeks later you get an epidemic of rheumatic fever, and unsurprisingly, therefore, all your M types of the rheumatic fever outbreak are the same. In the developing world populations, in places like the Pacific and Sub-Saharan Africa, we see incredibly diverse serotypes. And I think it's actually the opposite is true. I think it's that you're exposed to so many different distinct epitopes from different serotypes over childhood that drives some sort of abnormal immune response. And we just don't see the rheumatogenic strains in the settings where disease is endemic today.
Are there any more questions? If not, will you join me in thanking both David and Tom again?